Hello, today we're going to be talking about the digestive system. Some basic functions of the digestive system include takes in food, breaks down food to molecular-sized pieces, absorbs those molecules into the body, and eliminates waste products. Some components of the digestive system include the alimentary canal, which is also the gastrointestinal tract, the GI tract, which includes your mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum, which is a pathway by which food enters the body and wastes are discarded. The accessory digestive organs include your teeth, tongue, gallbladder, liver, salivary glands, and pancreas. Now, the way the digestive process works is you start with ingestion, which is pretty much putting food and drink into your mouth and swallowing it. Then comes propulsion, which is the swallowing part, which involves a movement called peristalsis, where the way the food goes down is like wave-like contractions of the muscle. Next is mechanical digestion, which is the chewing, mixing food with the saliva, churning the stomach. In this step, segmentation is being used in the small intestine. Then comes chemical digestion, which is the breakdown of the complex food molecules into basic components. Then absorption, which is when the components pass the GI tract into the blood. Then compaction, which is the formation of feces, and defecation, which is pooping pretty much.
Digestive system organs, relationships and structure. The first of the features is the peritoneum, which includes the visceral peritoneum and the parietal peritoneum. The peritoneal cavity is where you find serous fluid. The mesentery is a double layer of peritoneum that attaches the organs to the abdominal wall. You have intraperitoneal, which is inside the abdominal cavity, versus retroperitoneal, which is behind the abdominal cavity. You also have peritonitis, which is inflammation of the peritoneum. The bacteria found in the peritoneum will kill you, and this can also occur if you have a burst appendix.
Blood supply. Arteries of the celiac trunk. Arteries contained here include the hepatic artery, the splenic artery, and the left gastric artery, which contains the superior and inferior mesenteric arteries. Function of these arteries is to bring blood to the digestive system. Next is the hepatic portal system. Function of the hepatic portal system is to collect nutrient-rich blood from the digestive system organs and bring them to the liver. Histology of the alimentary canal. The first layer is the mucosa, or the mucous membrane. This is the innermost layer of the canal. Structures of the mucosa include the mucosal epithelium, which is made up of simple columnar epithelium, goblet cells, and cells that secrete hormones and enzymes. Lamina propria, which is areolar connective tissue under a layer of simple columnar epithelium. Also here are the mucosa-associated lymphatic tissues, or MALT for short. This is what Peyer's patches are made of, and their function is to defend against bacteria and pathogens. Next is the muscularis mucosae, which is a thin, smooth muscle layer creating folds in the small intestine, allowing for more surface area. Functions of the mucosa include the secretion of mucus, absorption of digested material, and protection against infection. The next layer is the submucosa. Wrapped around the mucosa, it contains nerves and blood vessels, and is made up of areolar and dense connective tissue. The next layer is the muscularis externa, which is made of smooth muscle. Structures of the muscularis externa include the inner circular layer, which is around the tube, and the outer longitudinal layer, which runs along the tube. Functions of the muscularis externa include segmentation and peristalsis. Peristalsis being the propulsion of food through the alimentary canal, and segmentation being the mixing and breakdown of food through the alimentary canal. The final layer is the serosa, or visceral peritoneum. Structures of the serosa are areolar connective tissue covered in a simple squamous epithelium. Function of the serosa is the secretion of serous fluid.
Next, we're going to talk about the enteric nervous system of the alimentary canal. The enteric neurons make up the nerve supply for the digestive system. The intrinsic nerve plexuses are found in the submucosa. There is the submucosal nerve plexus and then the myenteric nerve plexus. The submucosal nerve plexus is found in the submucosa, which regulates glands in the muscular mucosa. Then there's the myenteric nerve plexus, which is found in the muscular externa, which controls segmentation and peristalsis. Next is the extrinsic nervous regulation, which is done with the sympathetic and parasympathetic motor neurons, which is by activating the sympathetic nervous system, your digestive system slows down, and by activating the parasympathetic nervous system, it speeds up your digestive system and increases digestive activity. Next, we're going to talk about the functional anatomy of the digestive system. You have your mouth. General functions of your mouth include chewing the food, moistening with saliva, and swallowing. Next, we're going to talk about your lips and cheeks. The functions of your lips and cheeks are to keep food between teeth when you chew and help with speech. Some structures include the red margin, which is the red part of your lips, the labial frenulum, which is the fold that joins internal lips to gums, the vestibule, which is the area between your lips and cheeks and gums and teeth, your oral cavity proper, which is inside your teeth and gums where your tongue lives, your hard palate, which is the hard part of the palate, and then the soft palate, which is the soft part of your palate. Your uvula, which is the punching bag, closes off the nasopharynx when you swallow. And then the palatoglossal and the palatopharyngeal arches, they just anchor the soft palate to the tongue. And then the fauces anchors both of those arches. Next, we're going to talk about the tongue. The function of the tongue is to reposition food between teeth and mix food with saliva and form a bolus, which is basically a ball of food, and then moves it back into the pharynx. Some structures include the lingual frenulum, which is attached to the tongue to the bottom of your mouth, attaches the tongue to the bottom of your mouth, and then the papillae, which is the taste buds, the bumps on your tongue. There are 3 different types of papillae. There is the filiform, which don't have any taste buds in them, which is most of your tongue, and then there's the fungiform, which have taste buds, and then the circumvallae, which also have taste buds, but they're towards the back. And then there are the intrinsic muscles, which are contained within the tongue and not attached to the bone. They allow your tongue to change the shape. And then the extrinsic muscles extend to the tongue from bone and let you move your tongue in and out of your mouth. Next, we're going to talk about the salivary glands, which obviously produce saliva. Some functions of the saliva include cleans your mouth, dissolves chemicals in foods, moistens foods, has enzymes to start breaking down carbohydrates. Some histology. There are 2 different kinds of cells in the salivary glands. Serous cells, which produce a watery secretion that contain enzymes, and mucus cells, which make mucus. Next, we're going to talk about the extrinsic and intrinsic glands. The extrinsic produce most saliva in your mouth. You make about 1.5 liters of saliva. Then comes the parotid, which is made of serous cells. And then comes the submandibular, which is underneath the mandible. It has an equal amount of serous and mucus cells. And then we have the sublingual, which is underneath the tongue and mostly contains mucus cells. Next, we're going to talk about mumps, which is caused by a group of viruses, and it's a childhood disease. Next comes the intrinsic or buccal glands. They're little serous and mucus cells scattered around on the inside of your mouth.
The composition of saliva consists of water, salivary amylase, which breaks down starch, mucin, which is a protein that acts as an instant mucus, lysozyme, which kills bacteria, and IgA, which is an antibody. The control of salivation occurs when chemoreceptors and pressure receptors stimulate saliva production. The inhibitory factors are your central nervous system, which inhibits salivary glands, and dehydration, which is saliva production dropping. Halitosis is just another word for bad breath.
Teeth. Function of teeth is to aid in mastication, forming a bolus. Mastication being chewing, and a bolus being a ball of food that's swallowed. Dentition, which is teeth, refers to either permanent or deciduous teeth. Permanent teeth being adult teeth, and deciduous teeth, which are baby teeth, or teeth that fall out. There are 32 total permanent teeth, divided into 4 different types. First of these types is incisors, which cut food, canines, which tear and pierce food, and the premolars and molars, which grind and crush food. An impacted tooth is a tooth that stays embedded in the bone. A common example of an impacted tooth is a wisdom teeth that doesn't grow out. The standard adult dental formula is a set of 2 incisors, 1 canine, 2 premolars, and 3 molars on each side, multiplied by 2 to include the top and the bottom row, equaling a total of 32 teeth. Tooth structure. Gingiva, which are gums, help hold teeth in the alveoli, which are sockets in the mandible and maxilla. Parts of the tooth. First is the crown. Crown is the part of the tooth that sticks out. Next is the neck, which is in between the root and the crown. The root is the inner part of the tooth embedded in the bone. Next is the pulp cavity, which is a space inside of the tooth containing pulp. The root canal is a passageway from the apical foramen to the pulp cavity. Apical foramen is the hole where vessels and nerves enter the tooth. Periodontal ligament anchors the tooth to the alveolus, forming a gum phosis. Composition of the tooth. The first material is enamel, which is the exposed outer covering of the tooth. It is the hardest substance in the human body and does not repair itself. Next material is dentin, which is a bone-like material under the enamel. Next is cementum, which is a glue that attaches the tooth to the periodontal ligament. Finally, there's pulp, which is a soft tissue containing blood vessels and nerves. Clinical problems. Cavities, or caries, are bacteria in the plaque dissolving enamel and dentin, plaque being food debris and bacteria. Tartar, or calculus, is calcified plaque, which is caused by the buildup of bacteria. Gingivitis, which is pockets of infection in the spaces between teeth and gum causing inflammation. And finally, periodontal disease, which is tooth loss and destruction of teeth due to gingivitis.
Next, we're going to be talking about the pharynx. There are two different parts of the pharynx. There's the oropharynx and the laryngopharynx, which are both common passageways for food and water. Some histology. The pharynx is made of stratified squamous epithelial and mucus glands, and it is non-keratinized. Next, we're going to talk about swallowing. There are 2 different phases that are part of swallowing, which is the buccal phase and then the pharyngeal and esophageal phase. The buccal phase is where the tongue pushes the bolus of food back into the throat, and then the pharyngeal esophageal phase is when the tongue blocks off the mouth, the soft palate rises, closes off the narrow pharynx, the epiglottis covers the glottis, and the bolus of food moves back down into the esophagus. Next, we're going to talk about the esophagus, which is a tube that connects the mouth to the stomach. The esophageal hiatus is a hole in the diaphragm that lets the esophagus through. The cardiac orifice is when the esophagus connects to the stomach. The gastroesophageal sphincter or the cardiac sphincter acts as a valve to keep stomach contents from entering the esophagus. Some special features of the esophagus is the esophageal glands, which secrete mucus to help food slide down. Next is the longitudinal folds, which help with letting the food go down by expanding, in case you don't chew your food all the way through. The muscularis externa composition include The top third of the esophagus is skeletal muscle. The middle third is a mix of skeletal and smooth muscle, and the bottom third is just smooth muscle.
The gross anatomy of the stomach. You have the greater curvature and the lesser curvature, rugae, which are folds that allow for expansion, the cardiac region, the fundus, which is the dome-shaped top, and the body, which is the middle. You also have the pyloric region, which consists of the pyloric antrum, pyloric canal, pylorus, pyloric sphincter, and the duodenum of the small intestine. The mesenteries attach the stomach and other organs to the wall of the abdomen, and they include the greater omentum, the mesocolon, and the lesser omentum. The muscle layers of the stomach include muscularis mucosa, which has longitudinal and circular layers, and the muscularis externa, which has longitudinal, circular, and oblique layers.
Stomach histology. The tissue of the stomach wall is made of simple columnar epithelium lined in mucus cells. Mucus cells secrete mucus, protecting the stomach wall from stomach acid. Gastric pits are wells or depressions in the stomach wall. They lead to gastric glands, which produce gastric juice. Gastric juice is composed of hydrochloric acid and pepsin, which is a protein that breaks down enzymes. Types of cells in gastric glands. First are parietal cells, which make hydrochloric acid an intrinsic factor. Hydrochloric acid, or HCl, is stomach acid that breaks down proteins and activates pepsinogen, the pepsin precursor. Next is intrinsic factor, which allows for absorption of B12 in the small intestine. Recall that a lack of intrinsic factor causes pernicious anemia. Next are chief cells, or zymogenic cells, which produce pepsinogen when added with HCl, then produce pepsin and HCl, which together break down proteins.
Enteroendocrine cells. These are also called gut endocrine cells. First there's gastrin, which is produced by G cells. Stimulus for gastrin is the sight, smell, or thought of food, or food in the stomach. Targets of gastrin are the stomach and the small intestine. In the stomach, it causes increased HCl production, increased gastric motility, and increased gastric emptying. And in the small intestine, it causes an increased motility. Next is secretin, which is produced in the duodenum of the small intestine. Stimulus for secretin is acidic chyme in the small intestine. Targets include the liver, the pancreas, and the stomach. In the liver, it increases bile production. In the pancreas, it increases bicarbonate-rich juice production. And in the stomach, it decreases motility. Third is CCK, which is produced in the duodenum of the small intestine. Its stimulus is fatty chyme that enters the small intestine, and its targets include the pancreas, the gallbladder, and the sphincter of Oddi. In the pancreas, it increases production of enzyme-rich juice, contracts the gallbladder, squeezing out bile, and opens the sphincter of Oddi, releasing bile and pancreatic juice into the small intestine. Also here is histamine, which is produced by histaminocytes. Function of histamine is to help make more HCl. And finally, somatostatin, which decreases gastric juice production and motility. Stomach lining. The stomach is lined with mucus neck cells, which produce mucus. This mucus makes up the mucosal barrier. Mucosal barrier is a thick coating of high pH mucus that protects the stomach wall. Also in the stomach wall are tight junctions, which are between the cells of the stomach, forming a waterproof barrier.
Regulation of the stomach includes the direct control and the indirect control. The direct control uses the CNS, the central nervous system, with the parasympathetic and sympathetic. The parasympathetic uses the vagus nerve, which is cranial number 10, which increases digestive activity, and the sympathetic nervous system decreases digestive activity. Indirect control includes hormones such as gastrin, secretin, and CCK. Some phases of gastric secretion includes the cephalic phase. Sight, smell, taste, thought of food increases production of hydrochloric acid, enzymes, and mucus. In the gastric phase, food is in the stomach, which triggers stretch receptors, which increases juice secretion. Chemicals, the proteins act as buffers, which suck up hydrogen ions, increases pH, which causes an increase in hydrochloric acid and pepsinogen to bring the pH back down.
The intestinal phase of the small intestine. First up, you have excitatory, which is where chyme enters the duodenum. It stimulates intestinal mucosal cells to release intestinal gastrin. It also enhances gastric gland secretion. Then you have inhibitory, which is where the intestine fills with acidic chyme. It also activates the central nervous system, which causes the pyloric sphincter to contract so no more chyme can enter the duodenum. It also stimulates the release of secretin and CCK. Gastric motility and emptying. This is a response of the stomach to filling with food. The stomach stretches to accommodate the food because it is a smooth muscle that is easily stretchable, but it can also relax. You have receptive relaxation, which is before the food enters the stomach, and adaptive relaxation, which is where the food is already in the stomach and causes it to stretch. Second, you have gastric contractile activity. This is where the pacemaker cells set the basic electrical rhythm. Then you have regulation of gastric emptying. This is where the duodenum controls the rate of stomach emptying with secretin and CCK. Then you have 3 clinical problems: emesis, which is vomiting, gastritis, which is inflammation of the stomach wall, and gastric ulcers, which is erosions of the stomach wall.
General functions of the small intestine. After digestion is completed, both mechanical and chemical digestion take place. Mechanical being segmentation, and chemical being enzymes from the liver, pancreas, and small intestine. Small intestine is also where most nutrient absorption takes place, as well as propulsion, which is peristalsis. Gross anatomy. Structures include the plicae circularis, which are circular folds increasing surface area, as well as the mesentery proper, which connects the small intestine to the posterior portion of the abdominal wall. Found in the mesentery proper are blood vessels, such as the superior mesenteric artery and vein, lymphatics, which are lacteals, and nerves. Subdivisions of the small intestine. The first 10 to 12 inches is called the duodenum. Found here are submucosal glands which produce alkaline mucus protecting the small intestine from acidic chyme from the stomach. The duodenal ampulla, or hepatopancreatic ampulla, which is where the common bile and pancreatic ducts meet. And enteroendocrine glands, which produce secretin and CCK. The duodenal or hepatopancreatic ampulla controls the entry of bile and pancreatic juice entering the small intestine. Found here is the major duodenal papilla, which is a raised covering of skin around the ampulla, and the hepatopancreatic sphincter, or sphincter of Oddi, which is the actual muscle valve controlling entry of bile and pancreatic juice. The next subdivision is the jejunum, which is 8 to 10 feet of small intestine, making up the middle portion, which is usually empty. The final subdivision is the ileum, which is the last 10 to 12 feet. Here, more Peyer's patches are found, as well as the ileocecal valve, which is the end of the small intestine, regulating the passage of material from the small to the large intestine.
Histology of the small intestine. We got the plicae circularis, which are circular folds which increase surface area, and then villi and microvilli, which are finger-like projections that increase surface area. And then there are lacteals that carry absorbed dietary fats, and the capillary beds in each villus carry oxygen and removes nutrients. The wall of the small intestine, which is mucosa. There's the mucosal epithelial, which is simple columnar epithelium, which absorbs nutrients, with goblet cells, and enteroendocrine cells. There are also the intestinal crypts, which secrete intestinal juice, which is mostly water and mucus. The further you travel along the intestine, the fewer crypts there are and the more goblet cells there are. You keep adding mucus so the poop doesn't get stuck. Intestinal juice, mostly water and mucus. Panneth cells, which release lysosomes. Stem cells, which are rapidly dividing the cell that are replacing the worn out epithelial cells. Next comes the submucosa, which have Peyer's patches and Brunner's glands, or duodenal glands. The Peyer's patches, the further along you move in the intestine, the more Peyer's patches there are. And the duodenal glands, or Brunner glands, make the alkaline mucus. Next, we're going to talk about the regulation of the small intestine, which the parasympathetic nervous system and the sympathetic nervous system are used to control that.
Digestive processes occurring in the small intestine. The requirements for optimal intestinal digestive activity include bile, which break down fat, bicarbonate ions, which remove acid, digestive enzymes, which break down food, intestinal juice, and a slow delivery. The motility of the small intestine includes segmentation, which is mixing, peristalsis, which is propulsion, which also includes migrating mobility complex, which is waves of peristalsis that move down the small intestine. You have the gastroileal reflex, which is where the stomach is filled and increases peristalsis in the small intestine. And you also have gastrin, which opens the ileocecal valve. The large intestine. General functions include absorbing water and forming feces. The gross anatomy. The structures. First, we have the tenia coli, which is a band of smooth muscle that helps form the haustra. The haustra are the pouches that you see on the large intestine. Then you have epiploic appendages, which are just fat lobes that hang off the large intestine. Vermiform appendix is at the beginning of the large intestine and contains lymphoid tissue. The mesocolons are the mesentery sheets that attach the large intestine to the abdominal wall. Subdivisions include the cecum, which is the pouch at the beginning of the large intestine that receives things coming in from the ileocecal valve. Second is the colon. You have the ascending colon, the right hepatic flexure, transverse colon, left splenic flexure, descending colon, sigmoid colon rectum. It also includes the anorectal canal, which is the last part of the large intestine, the rectal valves, and the anal canal, which includes the internal anal sphincter and the external anal sphincter. Histology. The structures are the intestinal glands, which have goblet cells, and the anal sinuses, which secrete mucus. The tissues in the large intestine are simple squamous epithelium.
Bacterial flora. These are the gut bugs that function to produce vitamin K, vitamin B, and help in clotting. Digestive processes in the large intestine. Motility, which is movements, includes haustral contractions, which occur every 30 minutes, and mass movements or mass peristalsis, which only occur 3 to 4 times per day. Gastrocolic reflex is the stomach stretching, causing mass movements. Defecation. Defecation reflex occurs when feces is pushed into the rectum. Clinical problems. First are hemorrhoids, which is an inflammation of the hemorrhoidal veins. Next is diverticula, which are pouches or herniations in the large intestine. Diverticulosis occurs when there's not enough fiber and the pouches begin to tear. Diverticulitis occurs when the pouches are inflamed. Diarrhea occurs when not enough water is absorbed in the large intestine. And constipation occurs when there's too much water absorbed in the large intestine. And food poisoning includes salmonella.
So now we're going to talk about the accessory organs of the GI tract, which include the salivary glands and the liver. The liver helps break down amino acids, stores fat, and synthesizes proteins, stores vitamins and minerals, and removes nutrients from the blood. Some functions include metabolic regulation and hematological regulation, which processes bilirubin from the breakdown of red blood cells, forms plasma proteins, and produces bile, and finally, bile synthesis. Some gross anatomy include the 4 lobes, right, left, caudate, quadrate, falciform ligament, which separates left side from right side, round ligament, which hangs off the falciform ligament on the inferior portion, the porta hepatis, where the hepatic portal vein and the hepatic artery enter the liver. The right and left hepatic ducts bring bile out of the liver, which come together to form the common hepatic duct, which empties into the common bile duct, Some histology. Liver lobules are functional units of the liver. Hepatocytes are liver cells. Central vein is the hole in the middle of the lobule, which carries used blood out of the liver. And the bile caniculi carries bile to the bile duct. Next is the portal triad, which branches off into the hepatic portal vein, hepatic artery, and bile duct. The hepatic portal vein brings nutrients, the hepatic artery brings oxygen, and the bile duct removes bile out of the liver. The liver sinusoids is the area between the hepatocytes. The Kupffer cells are macrophages that help clean the blood out. The flow of blood through the liver is blood comes in from the hepatic artery and portal vein. They flow through the sinusoids into the central vein, and then the hepatic veins, and then into the inferior vena cava. Secretion and transport of bile. Stimulus for secretion is chyme, which causes CCK and secretin release. Transport of bile starts at the bile caniculi, into the bile ductules, right or left hepatic ducts, into the common hepatic duct, which will go into the common bile duct, into the duodenal ampulla, and then into the duodenum, or into the cystic duct, and then into the gallbladder.
The composition of bile includes bile salts, which are derived from cholesterol and emulsified fat, bilirubin, which is the yellow-green pigment from the heme of hemoglobin. This includes urobilinogen, which gives feces its brown color. Clinical problems include hepatitis A or B, which is inflammation of the liver, and cirrhosis, which can lead to portal hypertension, which is where the liver can't expand anymore. Jaundice is when you turn yellow from excess bilirubin. The gallbladder. The functions of the gallbladder are to store and concentrate bile. This is done through the use of a cystic duct, which connects the gallbladder to the common bile duct, and the sphincter of Oddi, which regulates the flow of bile and pancreatic juice in the small intestine. The stimulation of contraction is through CCK. Clinical problems include cholelithiasis, which is the formation of gallstones. The pancreas. There is the endocrine function, which has alpha cells which secrete glucagon, beta cells which secrete insulin, and delta cells which secrete somatostatin. The exocrine includes pancreatic juice. The structures include the head, the tail, the main pancreatic duct, and the accessory pancreatic duct.
Histology of the pancreas. First there are lobules, then there are pancreatic acini, which make enzymes and produce pancreatic juice. Islets of Langerhans are also here, which provide endocrine function. Composition of pancreatic juice. First there's water, which pancreatic juice is mostly made out of. Next are carbohydrases, which are enzymes that break down carbohydrates. Included here is pancreatic amylase. Next there are proteolytic enzymes, which are enzymes that break down proteins. Included here is trypsin, chymotrypsin, and carboxypeptidase. Next are lipases, which break down lipids. Included here is pancreatic lipase. Next are nucleases, which are enzymes that break down nucleic acid. And finally, electrolytes, which include bicarbonate ions. Regulation of pancreatic secretion. The parasympathetic nervous system is mostly in control, but there's also great influence from hormones. Hormones include secretin and CCK, which are synergists and work together. First, secretin from the duodenum is secreted when HCl gets into the duodenum, stimulating an increase in bicarbonate ions and potentiating the effects of CCK. Potentiate meaning to make stronger. CCK from the duodenum is secreted when proteins and fats enter the duodenum, increasing enzymes and potentiating the effects of secretin.
Next, we're going to be talking about the chemical digestion, what happens when you eat. As you can see, this is my burger snake, and it's broken down to show you how we digest things chemically. So first of all, in the mouth, salivary amylase breaks down starch and polysaccharides. And then in the protein, nothing happens. And then in the lipids, same thing, nothing will happen because protein and lipids cannot be dissolved in your mouth. Next, we stop digesting carbs while protein is broken down in the polypeptides by hydrochloric acid and pepsin, and again, nothing happens in the lipids. In the small intestine, polysaccharides get broken down into disaccharides by pancreatic amylase, and then the disaccharides get broken down even more into sucrose, maltose and lactose. Sucrose gets broken down by sucrase and then becomes glucose, and which is broken down to become glucose and fructose. Maltose is broken down by maltase, which is broken down to become 2-glucose. Lactose is broken down by lactase, which is broken down to become glucose and galactose. All three of those disaccharides are brush border enzymes, which they are produced in the microvilli in the small intestine. The polypeptides get broken down by trypsin, chymotrypsin, and carboxypeptidase, and they become amino acids. And then lipids are broken down by bile and the pancreatic lipase to become glycerol and fatty acids.
Absorption, which uses transepithelial transport. First, you have carbohydrates and proteins, which are both protein carriers in the cell. We have lipids, which use micelles, which diffuse into the epithelial cells, and chylomicrons, which are fats covered with protein skin. You have nucleic acids, which are protein carriers, and you have your vitamins. Vitamins A, D, E, and K are absorbed through micelles, and vitamins C, B, and B12 are absorbed through intrinsic factor. And then you have water, which simply diffuses.